हाजी क्या हाल है।
ठीक है
हेलो
जी। जुमा मुबारक। तुसा ॾियो।
सर, खैर मुबारक।
माशाअल्लाह वास्ते।
सुनाओ, तैयारी की थी कोई रमजान दी?
के तैयारी करनी है जी?
तैयारी मतलब ईमान
जी
की तैयारी।
ईमान। ईमान है इंशाल्लाह इंशाल्लाह। भरपूर इस्तकबाल थी। दुआई करो अल्लाह तौफीक देवे रोजे ने।
वो है ना, बिश्की के रह के तो ईमान पे ख्याल नहीं रख सकींदा।
बिस्की के चे ना पिछला माह रमजान ऑनलाइन ही ईमान तक कंट्रोल रह गया।
हाजी ऑनलाइन है तो कंट्रोल रही है।
जी
मैं के ऑनलाइन है तो कंट्रोल रही है ना।
जी जी ही बेया हाल वाल ठीक हो शुरू।
अल्लाह शुक्र, बिल्कुल ठीक। तुम सुनाओ।
ठीक हो खैर है। मैं माफ ऑफलाइन है। मैं लगता है क्लास नहीं है।
पिछले जुम्मे कहना थे। ये मैं
बिल्कुल नीला
बढ़ेती करें
कलर।
यहाँ पर।
पिछली दफा लेट है यही।
हाँ हाँ।
जले बता वाला स्लाइड सेंड की थी एक्सरसाइज पढ़ाया था।
ये सही मेहनत करनी पड़ी आपने उत्ते।
ये
वैसे पूरे एडम में क्या है, कसर नहीं छोड़ी, लेकिन ये खसूसी रहनी पड़ी है।
इस तरह तो आज को पता की दिखाना।
की ना
जलालाबाद था।
पता कीता है। आदन
क्या
जुलाई है,
दिन।
सब रब्ता करे जुलाई है, जून जुलाई है।
अच्छा
हाँ, जून जुलाई की सीट आसन उस लेवल।
हाथ हो रखसन।
हाजी ओ बॉय ई बहना थे।
कहीं को ओबा नहीं रहना ना।
ओ बवेसी है सफा। मैं आगे
स्पॉन्सर
में
करी होने।
5-7 सीटा चाहिए।
अच्छा क्या है।
अरे यार, आप रब्ता करना, मैसेज मैं कर दूंगा।
कर दूंगा तेरी जेब है जना।
हाजी ये सारे पैसे दिखे हैं।
जलालाबाद दे जरे मेन कंट्रैक्टर है ना वही था उधर संगवानी है।
अच्छा।
लेकिन है
सारी
काटे
गेम,
थे कब।
हाजी सारी गेम पैसे नहीं, देखो पता है।
गाल यह है काटे थे कब वो पैसे $2000 कंट्रैक्ट था 3000 मंगे ने कोई
ओए
पता
ओए,
कर देना कोई ईमानदार का।
मेरे एक दोस्त 5.5 लाख भरे हैं, पाकिस्तानी होता नहीं थी हाल तक।
ये तो जलालाबाद दा।
ना वो आई एच एस में आई।
अच्छा। हम्म।
5.5 लाख खवा बैठे थे, वो नहीं किया।
बोला क्या दल।
आधे ने 4 सेमेस्टर के बाद की थी।
कितने सेमेस्टर दे।
फोर तो बाद।
यार ये सिर्फ हमारी प्रैक्टिस क्लास है।
हाँ
हाँ
GM-13 की है बस।
ना करो, कुछ सुनाना पड़ेगा इसमें।
नहीं नहीं ये मम्मा तो बताई ये कौन है।
कौन है?
मामा तो
ये
बताइए।
देखना। हाँ
मोहम्मद तो बताई।
ये मैम तो
अपने
नहीं है
ग्रुप
कोई।
में इको अपने ग्रुप में स्पीड है। मैम केड़ी है?
All
पता नहीं।
ये माइक तो खुलिया भाई देखा।
students,
है?
वो मैम
यस
है।
मैम।
Assalamu alaikum.
वालेकुम
वालेकुम अस्सलाम।
अस्सलाम हाउ आर यू मैम
मैम
जुम्मा
क्या हाल है
मुबारक।
आपका? ठीक है? मस्तियाँ कर रही हैं?
मैम गुम थी ये बात।
बत्ती की क्लास खत्म।
ओए गूंगी तो नहीं गाड़ी ना क्या
कौन तेरे नाल शोर होने में क्या है?
Students. Hello, students.
यस मैम।
Now start practical classes. Rahmatullah.
मैम योर वॉइस इज वेरी लो, वी कांट लिसन।
Hello. Hello. Hello.
Hello.
You
यस
heard?
मैम यस।
You heard? Now start. Dr. Ali Muhammad Savar.
Yes, ma'am.
Do you question?
No, ma'am, I have no question.
What are lipids? What
No.
are
lipids
lipids?
are biological compounds which take part in the formation of biological systems. Example, cell membrane and other functions.
Muhammad, I wait
What's up?
you.
मैम योर वॉइस इज वेरी वेरी लो।
Oh, hello. Hello, students.
Yes. Yes, ma'am.
You heard?
Yes, ma'am.
Muhammad Sharas Haider,
Yes, ma'am.
question to you. Explain functions of lipids. Functions of lipids.
Great question, ma'am. Function of lipids,
Yes.
ma'am. Function of lipids. These are include storing energy, signaling, and acting as structural components of cell membrane. Lipids application in the cosmetic and food industries, as well as in nanotechnology. Energy storage. One of main functions of lipid is to energy storage. Cell structures. Lipids are present in every cell of human and are the main part of the cellular membrane. Hormones, progesterone, estrogen, testosterone, digestion.
Okay.
Chemical messengers role of lipids in body storage and prevention of energy, maintenance of temperature, membrane lipid layer formation, cholesterol formation, prostaglandin formation, and role in the inflammation.
Very good.
Yes,
Rahmatullah. Dr.
यस मैम।
Rahmatullah. Classification of lipids.
यस मैम।
Classification. Explain, please. Classification of lipids.
Lipids are classified at fatty acids and glycerides, and fatty acids control saturated and unsaturated, and glycerides are neutral glyceride and phospholipid. And third one is non-glyceride lipids and complex lipids. Non-glyceride lipids are waxes, sphingolipids, and steroids, and complex lipids are lipoproteins. And sphingolipids contain sphingomyelins and glycolipids. And um,
Okay. What are simple lipids? What are
yes,
complex lipids? Explain, please. And explain contain.
lipids, the lipid metabolites and simple lipids and
Mhm.
system based on fundamental lipids, simple lipids and complex lipids, ma'am.
Yes. Explain.
Main simple lipids are triglycerides, which are also known as triglycerols, steroid esters, and waxes esters. Uh, and hydrolysis of these lipids yields glycerols and fatty acids, and sterols and fatty
Okay.
acids, and fatty acids are called plus fatty acids, respectively.
Very good. Okay. Mm,
A compound lipid which contain
yes.
additional groups such as phosphoric acid,
Mhm.
sugars, nitrogenous base, or proteins included the phospholipid, glycolipids, and lipoproteins.
Okay. Thank you very much. Very good.
Thank you, ma'am.
Asmatullah.
Yes, ma'am.
Asmatullah, please. Explain, please. Explain of lipoproteins.
Lipoprotein, ma'am.
Yes.
Yes, yes.
Function of lipoproteins and classification.
Ma'am, lipoprotein is a biochemical assembly whose primary function is to transport hydrophobic lipid, which is known as fat, and molecules in water, as in the blood plasma or other extracellular fluid. There are four types of lipoproteins with on its characteristic protein and lipid composition. They are chylomicrons with very low density lipoproteins, which is known as LDL, and high density lipoproteins, which is known as HDL. The role of lipoprotein molecule is to transport fat molecules such as triacyl cholesterol, which is also known as triglycerides, and phospholipids and cholesterol within the extracellular water of the body, all the cells and tissues of the body. And lipoproteins are special particles which are made up of droplets of fats which are surrounded by a single layer of phospholipid molecules, and are molecules of fats which are attached to the phosphorus containing group. They are distinctive in being amphipathic, which mean that they have both polar ends and non-polar ends. Ma'am, there are 10 natural ways that, but this topic is not included in the.
Lipoproteins, for example, please. For example, chylomicrons,
Yes, ma'am.
chylomicrons. What are chylomicrons? Chylomicrons eat lipoproteins, yes?
Ma'am, high density lipoprotein.
Next.
Ma'am, then the normal values for the test of lipoprotein, which is known as LDL, they are less than 30 milligram per deciliter, and in many instances, people with have non-detectable levels of lipoprotein in their bloodstream. If the result is greater than 30 milligram per deciliter, it may indicate the risk of atherosclerosis, heart attack, stroke, and many other diseases. Uh, then we will say about the structure. As I described that there are two types of lipoproteins, low density lipoprotein which is called LDL and high density lipoprotein which is called HDL. So then in the lipids we will discuss about the cholesterol. Cholesterol is an insoluble in the blood and so it must be found to be lipoprotein in order to be transported. The LDLs and HDLs, uh, in LDL transport cholesterol from its site synthesis in the liver to the body cells where the cholesterol is separated from the low density lipoprotein, and then after that it is used by various cell purposes. But HDL, which is a higher density lipoprotein, it probably transport excess of unused cholesterol from the body tissue back to the liver, where the cholesterol is broken down into bile acids and bile salts, and then it is excreted. About 70% of the cholesterol in the blood is carried by low density lipids, lipoproteins, and most of the remainder is carried by high density lipoprotein. So if the density or of the concentration of the lipoproteins are above the normal, it may indicate many heart diseases, atherosclerosis, stroke, and many brain problems and stroke. So the body cells extract the cholesterol from the blood by many of the tiny codes, which is also known as receptor. They are on the surface of the cell. They bind with the lipoprotein, and then they attach to cholesterol. Uh, there are limits of the cholesterol in the body cell which are taken in. However, the cell capture a low density lipoprotein particle which inhibit the making of more low density lipoprotein. So these all were the functions of.
Hello?
Hello,
Mm-hmm.
ma'am, my internet connection was interrupted.
Yes. Very good. Very good. And answer Muhammad Sharaf Haider.
Yes, ma'am.
Muhammad Sharaf Haider. Please explain digestion of lipids. Digestion of lipids.
Ma'am, digestion of lipid, the digestion process has to break those large droplets of fat into smaller droplets and then enzymatically digest lipid molecules using enzymes called lipase. The mouth and stomach plays a small role in this process. But mostly enzymatic digestion of lipids happen in the small intestine. Hence, ma'am, the digestion of certain fats begins in the mouth where short chain lipids break down into the diglyceride because of the lingual lipase. The fat present in the small intestine stimulates the release of lipase from the pancreas and bile from the liver enables the breakdown of fats into the fatty acid.
Okay.
After the
Question.
fat has been digested, fatty acids are passed through the lymphatic system and then throughout the body where you are bloodstream to be used or stored for the energy, cell repair and growth, and for lymphatic system also absorb fatty acid to help infection. The major products of lipid digestion is fatty acids and 2-monoglyceride enter the enterocyte by simple diffusion across the plasma membrane. A considerable fraction of the fatty acids also enters the RBC via fatty acid transport proteins in the membrane. Uh, there are enzymes, lipid metabolism, and enzymes in liver. The lipid is food as many TG and here a small amount of PL and CH. In the small intestine, bile acid and pancreatic enzymes include pancreatic lipase, phospholipase, A2 cholesterol, esterase, etc. In bile, hydrolyze lipid into free fatty acid. Digestion of fats takes place in the small intestine. Fats reach the small intestine in the form of large globules. The liver releases bile juice which emulsifies the fat. It breaks down the large globulus into the smaller globulus. Lipase act as and break down the globulus and molecules. In the duodenum, bile starts break up lipid droplet into smaller droplets. Emulsification allows pancreatic lipase digestion, triglyceride into fatty acid, and monoglyceride and glycerol. They break down triglyceride form. Hello, ma'am. Hello.
Muhammad,
Yes, ma'am.
Muhammad, sir, explain this process of saponification.
What, ma'am? Emulsification?
Emulsification.
Okay.
Sponification,
Correct answer.
sponification.
Correct answer. Yes, okay, very good.
क्या किस ma'am?
And saponification process.
Sponification, साबुन बढ़ता जरा.
Saponification.
Parang dasing SO.
Soap formation, soap.
Yes.
Soap?
Muhammad Sharaz, very
What, ma'am?
good. Uh-huh. Very
Soap
good.
formation?
Saponification.
Yes, ma'am.
Very good.
Okay, ma'am.
Rahmatullah, help
हाँ,
please.
soap.
Saponification process.
Saponification.
Simonification.
Spoon lecture spoon चम्मच दीवाल उन्दिया को lecture fication sponification.
Saponification.
Okay, ma'am. Sponification is the process that involves the conversion of
हाँ
fat,
ब्लैक
oil,
चुप
or
करके बैठ तू
into soap and alcohol by the action of heat in the presence of aqueous alkyl, for example, and sodium hydroxide. Soaps are salts of fatty acid, and fatty acids are monomers of lipid that have long carbon chains as least 10, for example, sodium palmitate.
Saponification is
Saponification is the hydration reaction where free hydroxide breaks the ester bonds between the fatty acid and glycerol of a triglyceride, resulting in free fatty acids and glycerol, which are each soluble in aqueous solution.
products. Which products? Glycerols, yes. And
Yes, ma'am.
and
Emulsification, ma'am, product.
Yes.
alcohol, soaps,
Alcohol. Yes, yes, yes. Okay. Two products. Okay.
yes, ma'am.
Two product. Two product. Molecules. Glycerol and
Alcohol,
soap. Soap.
soap, yes, ma'am.
Soap. Eat salt. Okay.
Okay, ma'am.
Mhm. Very good. Uh, Zishan, Dr. Zishan,
Yes, ma'am.
Thank you, ma'am.
thank you.
बेटा मैम
Dr.
तो खुश कर रही हैं
Zishan,
सबको डॉक्टर बुला के।
मैम दिलूश कर दिया आपसे। Yes, ma'am.
Zishan, please. Degradation process, degradation, triacylglycerols, explain.
Ma'am, degradation of triglycerides.
Yes, triglyceride.
Ma'am, first of all, triglyceride is formed by the 3 units of monomers of glycerol. 1 second. यार ये चाल नहीं डाला है।
Zishan.
Ma'am, 1 second. There is a network problem. I speak. 1 second.
Think, please. I wait you.
Yes, ma'am. Degradation of triglycerides, ma'am. Ma'am, when the body requires fatty acid as an energy source because lipids and fats are the high energy source for our body. And hormones, glucagon break down the triglyceride by hormone sensitive lipase. Triglycerides are broken up with the help of hormone sensitive lipase enzyme. And these lipase enzymes are into the free fatty acids. These triglycerides are breakdown into free fatty acids. Special enzymes on the walls of blood vessels called lipoprotein lipase must break down triglyceride into free fatty acids and glycerol. Hormone sensitive lipase and lipoprotein present on the blood vessel walls. These lipoprotein lipase break down triglyceride into fatty acids and glycerol and obtain energy from them and utilizing the different metabolic pathways of body.
Okay. Okay, very good. Muhammad Zafar.
Yes, ma'am.
Muhammad Zafar, please. What saturated fatty acid and unsaturated fatty acid? What?
Saturated.
Yes.
Saturated.
Explain structure. Fatty
Mhm.
acid, yes.
Yes.
And what are saturated fatty acid and unsaturated fatty acid?
Okay. Saturated fatty acids are derived from both. They have only single bond, and unsaturated fatty acids has double bond in their structure. The
Yes.
saturated acids are derived from both animals and plant soil, and resources of saturated fatty acid include butter, meat fat, tropical oils like palm oil, coconut oil, uh, and they have straight chain in their structure and even number of carbon atoms. And the unsaturated fatty acids, there is at least one double bond, and they are usually derived from plants.
Okay. For example, fatty acid.
Example of which fatty acid, saturated or?
Yes. Saturated and unsaturated.
Okay. Example of is, for example, stearic acid, like that, saturated acid, stearic acid, palmitic acid, or uric acid. These are saturated acids, and unsaturated acids is like oleic acid, soybean
Okay.
oil, canola oil.
Okay. Uh, explain, please. Iodine number.
Which number? Iodine number.
Iodine number. Yes.
Okay.
Explain.
Yes, yes. Iodine number is a measure of degree of unsaturation of oil or fat or any wax. The mean any fat oil which is about which degree is which is about which at which degree is unsaturated. The amount of it is amount of iodine in grams that is taken up by 100 gram of oil, fat or wax. And iodine number is usually show us the unsaturation of any oil or fat. And saturated oil, fats, waxes takes up to no iodine, therefore their iodine value is zero. But unsaturated oils, fats and waxes take up to iodine, and unsaturated compounds contain molecules like double bond and triple bond, which are very reactive towards iodine. And the more iodine is attached, the higher is the iodine value. The more reactive and less stable, softer and most susceptible to oxidation and reduction is that oil or any fat or wax. In performing the test, a known excess of iodine, usually in the form of iodine monochloride, is allowed to react with the known weight of that oil or any fat. Then the amount of iodine remaining unreacted is determined by titration.
Okay.
Drying oil used, yes, ma'am.
Muhammad Safar. Uh,
Yes, ma'am.
iodine numbers shows the number of double bonds. Yes.
Yes, ma'am. Unsaturation.
How much double bonds? How much double bonds? Okay, unsaturated. Very good. Thank you very much. Next, please, individual work. You can see questions.
Ma'am, unsaturated fatty acid do not have any double bond.
Unsaturated fatty acid have double bond,
अच्छा
double or triple bond. Saturated के पास single होते हैं.
अच्छा सैचुरेटेड होते हैं ओके मैम
Unsaturated
बस
is
यही इंफॉर्मेशन रह
double
गई थी
bond.
मैं
Yes.
एमबीबीएस में हासिल करने के लिए, बाकी हमें सब पता था.
जी भाई
Doctors, unsaturated. What unsaturated fatty acid? Unsaturated fatty acids have double bonds. Okay.
ओके मैम
Double bonds.
यस मैम
Yeah, iodine numbers shows the number of double bonds. Okay.
Double unsaturation. Yes, ma'am.
Iodine number. Okay. You can see questions. You see questions.
यस मैम.
Yes, ma'am.
Write, please. Questions. Write, please. students?
Yes, ma'am.
Prepare answer to questions.
First one.
No, 4 questions. Everything questions.
All questions we
All
have to answer.
questions, all questions. You are ready?
Yes, ma'am.
Ma'am, these are our home tasks we have to do right now.
All questions.
अच्छा ये क्या कह रही है?
तो भाइयों से पूछ
All
लेना
question अभी के अभी
सुनाने हैं
सारे
लिखने
सुनाने हैं।
कैसे सुनाने हैं इसको? पागल हो गई है?
बारी बारी सबने।
Give you time?
Ma'am, very less time for these questions.
How much time?
Ma'am, these are too much for us right now. We can prepare them in the next class.
Next
We cannot
class?
prepare all the questions right now.
Okay. Correct answer. Prepare answer next class, yes?
Yes, ma'am, in the next class.
First,
Next class. Okay.
भाई मुझे छुट्टी दो।
तू छुट्टी रह के मेरे पास आना ही।
फर्क कौन है?
Students, I wait you next class answers.
Okay, ma'am. रहमत
बस
इससे
ठीक है।
क्वेश्चन पकड़ ले। रहमत क्वेश्चन पकड़ ले इससे। लीडर
Okay.
ग्रुप में पकड़ ले।
Today,
स्क्रीनशॉट लिया हुआ है, सेंड कर दूंगा।
बस ठीक हो गया।
बस 10 विधानियां करो तो
today,
मिलूंगा।
no. Ali Muhammad Zafar.
Yes, ma'am.
Very good. Excellent.
Thank you.
And Muhammad Sharas Haider.
Yes, ma'am.
You answer yes?
Yes, all question, ma'am.
Very good. Very good. Asmatullah. Very good. And Zishan.
Yes, ma'am.
Zishan, very good.
Oh,
Nishan
ma'am.
mark is the
Okay.
mark to Nishan mark.
I also answered, ma'am. I also answered.
Rahmatullah, very good. Zishan, very good. Rahmatullah, very good.
Very
And Ali Muhammad Zafar, yes?
yes, ma'am.
Yes, ma'am.
Yes.
So very good. Okay, goodbye. See you
Goodbye.
next week. See you next week.
Thank you,
Inshallah,
ma'am.
ma'am.
I
Inshallah.
wait, inshallah. I wait answer.
Okay.
Okay.
Just with that.
Ma'am, I want to say something.
वह चबले ना मार यार। वो वे नहीं है नॉन नॉन है क्या है?
तैं पानी ना उड़े तू जो छिता पे थे।
ना तो संग
यार
जितना
उड़ा
होने
दे, कह
होती।
रही थी अटेंडेंस लगवा दे।
आज दिशा में हम इकट्ठे हैं। आज मैं
यार-
वैसे वॉशरूम।
यार रैम अदाबिया कह रही थी कि अटेंडेंस लगवा दे।
लीडर को कहना।
लीडर
वो
क्या
मैं
है
टेक्स्ट कर दूंगा।
बस ठीक है। ओके मैम, до свидания।
До свидания. Get out of the
मैम,
Zoom. Yes?
मैम, our
Yes?
classmate Rabia, ma'am, her grandfather passed away today. That's why she's absent.
Rabia?
Yes, ma'am.
Uh, mhm. Okay.
राबिया सादिक।
Other?
Her grandfather passed away today, ma'am.
Okay. Mm-hmm. Understand. Mm-hmm.
That's why
मैम
she
कैन यू
could not attend to class.
so can you
Okay.
put her present today?
Okay.
एप्लीकेशन वो एप्लीकेशन।
वह
Class
हाँ
के
वह तू चुप कर बे बुलट यार।
भोंकते ना।
Дишан, до свидания.
ओके, до
До
свидания
थैंक यू
свидания.
मैम।
До
इतना
До
свидания।
свидания. Ага, до свидания.
Thank
मैम तुझा लीव करो सा बट्टा ले।
Вот.
बड़ा
कोई
अच्छा बात।
कोई भी नहीं जा रहा है।
Ma'am, we are discussing our study plan with each
Okay.
other. That's why we are not leaving.
Okay.
उसको
I
होस्ट
will.
बना रहे ना देख। होस्ट बना रहे।
I will. It's Moodle. Okay.
Okay.
ओके मैम।
Then.
ओके मैम, до свидания।
अबे
जल्दी।
हाजी गिरेगा हाजी।
अच्छा, आप एक गला शुरू करो, मैम ने यहाँ पे चल जाना।
हाजी तेरे मुंह झा खा गए ना हाजी।
वो रहमतुल्लाह नवी मीटिंग बना के नो वाला ज्वाइन कर गिनते हैं।
यार उसमें सब ने नहीं आना यार, साढ़े जो कोई कदा गैप हो जाना यार। नोफल